All right, today we are moving on to your notes that are labeled Day 8 DNA replication, DNA, cell division, genetics, stuff like that. That's the thing that I find most interesting. Not that cellular respiration and photosynthesis aren't really important. It's just kind of my bread and butter is DNA and everything that goes along with that. So I'm really excited to finally begin this little unit here. So you have a list of 1, 2, 3, 4, 5, 6 names. You don't need to write down every single thing about it. I'm going to tell you under each name what to write down. Frederick Griffith, if you can look at your handout, it says Griffin. It looks like it defaulted to Griffin with an N. So if you can please cross that out and put TH. His name is Frederick Griffith. So he did this really cool experiment. So believe it or not, guys, there was a point in time where people did not know what the hereditary material was. They didn't know that DNA is actually what gets passed down to offspring and blah, blah, blah. So this guy did this experiment. And so kind of follow along with my cursor here. You have bacteria. And this is living S bacteria. And what he did was he injected mice with this living bacteria and the mouse died. So what we would assume is that this bacteria is pathogenic. Pathogenic meaning disease or death causing. All right, move on to the second little scenario here. Living R cells. Once again, this is just a certain kind of bacteria. He injected it into the mouse and the mouse died. So what we would assume is that living R cells are non-pathogenic, not causing disease or death. Third little experiment he did, he took this first one and he heated them up and killed them. The first bacteria sample that he injected in the mouse, remember the pathogenic bacteria, he heated it up, killed it, injected it into the mouse, and the mouse didn't die. And that, okay, cool, you took something that was pathogenic, killed it, and then it no longer worked. All right, that's what we would assume to happen. The fourth thing that happened right here is what's actually really interesting. He took these two, the second one right here, non-pathogenic living, pathogenic dead. Individually, neither of these killed the mouse, right? But when he mixed them together, the mouse ended up dying. And that's pretty cool and that's really interesting. So what happened? Why was that? So his conclusion is that the living R non-pathogenic bacteria transformed into the pathogenic bacteria by an unknown heritable substance. So bacteria actually do this thing where they transfer DNA between each other, or if a bacteria cell dies, a bacterial cell near it will it's almost like robbing it, I guess. Well, I guess it's dead, so it doesn't matter. It's like, yeah, it takes its DNA in. And so that's what the R bacteria did, is it took the pathogenic DNA of the S bacteria into itself, making itself actually pathogenic. So that's really cool. So basically, what you would write down under his name is, there is an unknown heritable substance. That's really all you would need to write down under his name. There is an unknown heritable substance. Oswald Avery, all you need to write down under him was that he discovered that the unknown heritable substance was DNA. So discovered that the unknown heritable substance was DNA. And so these two guys right here are actually talking about bacteria and the transfer of DNA between bacteria. So they didn't discover DNA itself, they just discovered between bacteria, this is the heritable substance. All right, Hershey and Chase, 1952. What they did was they wanted to figure out the virus, which is called a bacteriophage, that infects bacteria. So you didn't know, you might not have known, bacteria can actually get sick. Viruses do infect bacteria. They're called bacteriophages and they look super crazy, like something you would find in outer space. And it was known that these viruses injected something into the bacteria, but they didn't know. So this is a picture of a bacteriophage actually injecting something into the bacteria. They didn't know what it was. So they wanted to figure out, are these bacteriophages injecting sulfur? Are they injecting phosphorus? Or are they injecting protein, or are they injecting DNA? And how they figured that out is that they radioactively labeled the protein of the bacteriophage, and they radioactively labeled the phosphorus of the DNA in the bacteriophage. And so if the bacteriophage is injecting protein, you'll see sulfur light up inside of the bacteria. If it's injecting DNA, then you'll see phosphorus light up inside the bacteria. And so to make a long story short, if you looked at this, so the sulfur experiment is the pink one on the top. The phosphorus experiment is the blue one on the bottom. And what they found is that phosphorus ended up inside the bacteria. Why did they choose sulfur and why did they choose phosphorus? Because you find sulfur in proteins and not in DNA. So if sulfur had shown up, they knew that that would be the protein. Why did they choose phosphorus for DNA? Because DNA has phosphorus and proteins don't. So if they found the radioactively labeled phosphorus inside the bacteria, then they know it had to be DNA. So they figured out, okay, even viruses, so bacteria are transferring DNA among each other. And then in this experiment, this is what viruses are injecting, depending on the virus, into the bacteria. So the conclusion, what you would write down under Hershey and Chase is, viruses inject DNA into bacteria. DNA must be the genetic material. So all of these experiments are just like confirmation that genetic material is DNA, basically. That is like the genetic material. All right, Chargaff, Edwin Chargaff. He found that even though DNA composition varies between species, the percentage of A equals the percentage of T. And you're like, what is A and T? What are you talking about, lady? Adenine, thymine, guanine, and cytosine, those are the nitrogenous bases found in nucleotides. So remember, a nucleotide is the monomer that makes up nucleic acids. And so a nucleotide is made up of a phosphate, a sugar, and a nitrogenous base. Well, these are the four options for that nitrogenous base. Depending on that nitrogenous base that's there, that gives DNA its code. All right, and so he found that A equals T, C equals G. So therefore, since DNA is double-stranded, if there is an A on one strand, it will be bonded, hydrogen bonded, to a T on the complementary strand. All right, on the complementary strand. Okay, so basically he found that A pairs with T, C pairs with G when we are looking at DNA, double-stranded DNA. All right, Rosalind Franklin, the only thing you need to write down about her, I wouldn't write down any of this stuff, except she was the first person to actually get X-ray images of DNA. So she took X-ray images of DNA. She didn't get credit for this until after she died, which is unfortunate because these guys, Watson and Crick, they took her X-ray images and then they got credit for coming up with the double helix. And they all ended up getting credit for it, but it was just, you know, it's just the way it was back then with female scientists and male scientists. The male scientists were more likely to get the credit for stuff. And so Watson and Crick, what you need to write down under them is they used Rosalind Franklin's X-ray image of DNA to come up with the model of DNA, which is a double helix. All right, so let's talk about DNA structure. So DNA is a double helix. The backbone of DNA are the sugars and phosphates. So if you look at just this thing right here, so look at my cursor, you have a phosphate, a sugar, and a nitrogenous base. And these are the four different options for nitrogenous bases. So these are the four different types of nucleotides. They are bonded together here. So you can see the sugar and the phosphate. So these are four nucleotides bonded together, making up a strand of DNA. And so the backbone of DNA is the phosphate and sugar. And it's kind of like the rungs, like if this was a ladder, because you have two of them. And I have a really good picture later on. The rungs of a ladder. So if you have a backbone over here, and then you have the complementary strand on the right side, you'd have the backbone over there, and then you'd have the base pairing occurring in the middle. The nitrogenous bases, like I said before, are adenine, guanine, cytosine, and thymine. Don't write this down. You don't need to do purine and pyrimidine. I don't care if anyone knows that. Even in AP, I don't care if you know that. There's like, okay, adenine and guanine are double ringed and thymine and cytosine are single ringed. Like, who cares? I'm sure somebody does, but not for this class. So Adenine and thymine are going to base pair. Guanine and cytosine are going to base pair. Once again, I had said that earlier under Chargaff's rule, but this is just giving you the picture for it. Okay, so here's a really good picture of DNA. This picture right here doesn't give you the complete view of what DNA actually looks like. This one gives you a good view of what DNA actually looks like. So here's A is going to show you key features of the DNA structure, very simplistic model. B is going to show you kind of the chemical structure, and then C is going to show you the space filling model, it doesn't give any letters or anything like that. It's just kind of showing you the clouds, the atoms and their clouds and stuff like that. So here's what I need you to know. I need you to know, kind of like what I said before, that one nucleotide over here is going to base pair with another nucleotide over here. These are the two strands of DNA. And that between two nitrogenous bases, you have what's called a hydrogen bond. And they're going to hold the molecule together like a zipper. Hydrogen bonds are strong attractions, but they're weak bonds. That's why they're represented by a dotted line, because they're that weak. There really are just a strong attraction between a partial positive and a partial negative charge between two different molecules. But I need to write down they're held together by hydrogen bonds, and the shape of DNA is a double helix. And so what causes the double helix shape? What causes the double helix shape is the anti-parallel nature of the two strands. So what I'd like for you to write down is the two strands run counter to each other. That's what anti-parallel means. They run counter to each other. One strand, the phosphate end is up on the nucleotide. On the other strand, the phosphate group is down. So these nucleotides are turned upside down. And then I'd also like for you to write down the 5' end. 5' just refers to, if you look at my cursor here, you see where the oxygen is? If you count the carbons to the right of the oxygen, 1, 2, 3, 4, 5, the phosphate group is hanging off of the 5th carbon. And then it's the same thing here. If you look at the hydroxyl group, the OH, it's 1, 2, 3. It's hanging off of the 3rd carbon. So you're going backwards since that's upside down. So the phosphate group is on the 5' end, the hydroxyl group is on the 3' end, and anti-parallel means that the two strands are running counter to each other, and because of the negative charges on the phosphates, it's going to cause a wrapping. So the anti-parallel running counter to each other is going to cause like a wrapping, and that's what the double helix comes from. Okay, DNA is, and this is in eukaryotes, so write eukaryote on top of here, right? Eukaryote. In a eukaryote, DNA is wrapped around proteins called histones, and that's how it's packaged. And then I'd also like for you to write the word chromatin at the bottom. Chromatin. And I've mentioned that when I talked about the nucleus the other day. So DNA is wrapped around these proteins, and it's packaged in DNA with its associated proteins, and those proteins are called histones. It's called chromatin, right? And eukaryotes have that. Chromatin, right before a cell divides, is going to further condense into chromosomes. And you don't need to write anything down about this slide right here. The only thing I want to say is that I'll just talk about that a little bit more when we do mitosis and cell division. All right, let's compare DNA between prokaryotes and eukaryotes because there is a big difference. Remember, prokaryotes are your bacteria that don't have a nucleus. They're very simple, no membrane-bound organelles. Eukaryotes do have a nucleus and do have membrane-bound organelles. The one thing they do have in common is that DNA is double-stranded in both. You wouldn't believe how many times students will tell me prokaryotes have single-stranded DNA. No, and I think they misinterpreted what the question actually was, and I'll tell you in a second what I think they thought. DNA is double-stranded no matter what. It's always double-stranded. In prokaryotes, DNA comes in a circle. The chromosome is in a circle. And in eukaryotes, the chromosome is actually linear. In prokaryotes, they only have one chromosome, and that's what makes it circular. There's just one circular chromosome. Eukaryotes are usually going to have more than one, so more than one chromosome in eukaryotes. The DNA of prokaryotes is found in the cytoplasm, and in eukaryotes, it's found in the nucleus. And then eukaryotes are going to have DNA wrapped around histones. Prokaryotes have no histones. And DNA with its proteins is called chromatin. And then in prokaryotes, it's just supercoiled DNA, just DNA by itself. So if you need to keep writing, go ahead and pause because I'm going to move on. It's important to know those differences. Let's talk about DNA replication. So DNA needs to be copied before a cell divides. It needs to be copied because the new cell needs the same DNA in it, right? So we are going to go through how DNA gets copied. Cross out the question that says who proved the semi-conservative model. It was these guys called Meselson. I don't even remember the first guy's name. I feel bad. Meselson and Stahl, and they just proved this, what's called the semi-conservative model. So you can cross their names out. I'm not going to ask you their names, or you can cross the question out because I'm not going to ask you their names. But it's important to know what semi-conservative means. So on A, your parent molecule, that's also known as your original. So I would write original underneath that. So your original DNA, and as you can see, one strand is complementary to the other strand. A goes with T, C goes with G, T goes with A, T, C, G. So if I gave you this left side right here, you should be able to tell me what is going to be on the right side. Okay, so this is your original DNA. It gets separated, and each the cell is dividing, and so each new daughter cell gets one strand that was from the original and one new. One strand that's from the original and one new. And so that's what's called semi-conservative, is that the daughter DNA molecules each consist of one of the parent strands and one of the new strands. DNA replication is kind of complicated. I'm going to try to simplify it for you, and all I'm doing by showing you this picture is just trying to convey how complicated it can actually be. So what I'd like for you to do is just listen to the steps before writing them, and then we're gonna go through each one in detail, and you can write it then. So the first step is that DNA gets unwound with an enzyme called helicase. So just listen, don't write right now. DNA gets unwound from an enzyme called helicase. Initiation proteins are going to separate the two strands, and it forms a replication bubble. I'm going to show you pictures of all of this. Primase is an enzyme that puts down an RNA primer. DNA polymerase is an enzyme that's going to add all of the new bases. It's basically forming that new DNA strand. You have a leading strand and a lagging strand, and I'm going to explain what leading and lagging strands are in Okazaki fragments. Then DNA polymerase 1 is an enzyme that comes in and replaces that primer from step 3. And then DNA ligase is an enzyme that seals the fragments together. All right, so that's kind of complicated, right? All right, so now you're going to start writing. Here are the steps of DNA replication. Step 1. So steps in replication. Step 1. Helicase unwinds DNA at the origins of replication and creates a replication fork. So go ahead and pause so you can write that down. And I'm going to move on. So if you're looking at this picture, you're following it down. All right, you're following it down. This is a process, like a multi-step process. Here is just your normal DNA, parent strand of DNA. At various points on that DNA strand, helicase comes in and helicase unwinds and pulls it apart. Pulls it apart. And then step 2, under step 2, write down Initiation proteins separate the two strands to form a replication bubble. So step 2, initiation proteins separate two strands to form a replication bubble. Go ahead and pause if you need time to write because I'm going to move on. All right, so you got step 1 and 2 written down. Helicase is going to unwind DNA and the proteins are going to pull it apart. What I need you to write down or just notate is that this whole thing right here, this round thing, is called the replication bubble. And notice that DNA is getting replicated in both directions. This is an efficient process. As you can see, DNA is not just getting replicated at one point on a chromosome. Helicase comes in and it's very efficient. It's going at different places. And what you can see here in this box, this is the fork. Okay, so I need you to know bubble, and then that's the fork. And so DNA is getting replicated in each direction at the fork. And if you follow it down, there you go, it's getting replicated. Eventually those replication bubbles are going to meet each other, and you're going to end up with two separate daughter DNA molecules. All right, step 3, Step 3, primase adds RNA primer. Primase adds RNA primer. So here's the way I think of it. The enzyme that's going to make the new DNA strand complementary to the old one needs some encouragement. Kind of like if there's an event going on and you want celebrities to come to the event, they usually will roll out a red carpet and they'll go on the red carpet, they'll get attention and the paparazzi will take pictures of them, whatever. This is how I think of primase and RNA primer and DNA polymerase. Primase comes in because DNA needs to get copied, but DNA polymerase is not going to come copy DNA unless it's encouraged. And so what primase does is it puts down an RNA primer. What that means is, remember, RNA has a uracil instead of a thymine, and uracil is a nitrogenous base. It's considered really reactive and has charges and stuff. And so this RNA primer is kind of like rolling out the red carpet. Then DNA polymerase is going to come in and start adding actual DNA. We're not trying to get RNA, we're trying to get DNA, but you have to put down a little piece of RNA to encourage DNA polymerase 3 to come in and start adding new nucleotides. All right, so that's step 4. DNA polymerase 3 adds nucleotides in the 5' to 3' direction. What that means. So pause to write that down because you're going to want to hear this. What that means is DNA polymerase cannot add a new nucleotide on the phosphate side. So it can't come right here. Phosphate's too big and bulky and annoying, and it just can't. It just can't. That's the 5' end, so it's not going to add new ones here. It's going to add new nucleotides to the hydroxyl end, which is the 3' end, okay? And then what I also want you to notice from this picture are the 3 phosphates here. Remember, when 3 phosphates are together, that's a lot of potential energy. And so all I really want you to know is DNA replication takes energy. It takes energy. All right. I'm going to move on. Okay, I need to talk a little bit about the leading and lagging strand because this is going to come up next. All right, leading and lagging strand. So here's our origin of replication. This is where helicase has come in and has unwound DNA, and binding proteins have opened it up right here, right here. So right here in the middle of the bubble is where replication starts. It can only go to the left here, and it can only proceed to the right here as DNA is opening up. And the reason is because it can't move in this direction, and this one can't move in this direction because DNA polymerase cannot add to the 5' end of a nucleotide. This is a very difficult concept for students to understand. It took me a long time, past high school, past my first year of college, to actually understand what this means. And since this class is so condensed, I just want to show it to you and say, there is a leading strand that goes in this direction, and then there's a backtracking that happens behind it called the lagging strand. And those lagging strands are also known as Okazaki fragments. So for AP, this is something that you would have to actually conceptualize and really work at understanding. And what I can do, hopefully we're in the classroom by this time, but for that class in particular, I would really emphasize that DNA polymerase can only add to the 3' end, can't add to the 5' end, so it can only replicate on the top. It can only go this way. On the bottom, it can only go this way. And if it starts right here, then it will have to come behind itself like that and replicate like that. So hopefully that made a little bit of sense. I'm curious to know who actually understood what I just said. Maybe I don't know. I probably want to ask in the Zoom call, like, who actually understood it? But I might ask for a volunteer. Is there anyone that would attempt to explain Okazaki fragments to us? And if everyone says no, then I'll just move on and whatever. But I would like for someone to make the attempt. That would be really nice. So this lagging strand, these backtrack, little short bursts, little segments are called Okazaki fragments. And if you notice, primer gets put down and polymerase comes in and makes new DNA. Primer gets put down. It's like a backtrack as the fork opens, okay? So the problem here is look at all these little red spots. We don't want red spots. We want only blue. We don't want RNA in our DNA. We want only DNA. So what happens is, step 6. What happens is step 6. Let me see what I wrote down. DNA polymerase 1 replaces the RNA primer. So write that down on step 6. And then 7, DNA ligase seals the fragments together. So pause if you need to write those because I'm about to switch this. All right, so the problem is we have all these red dots and we want only blue. So DNA polymerase 1 comes in, replaces the red with blue. It replaces RNA with DNA, and then ligase comes in and seals it all together. So you end up with your beautiful, at the end, your 2 new strands of DNA. And so here's a picture. What I would do if I were you, I would go back and I would go through each step and I would number where you see it happening on this picture. I would number where you see it happening on this picture. All right, at this point in time. So I would pause and do that. Okay, and then I have here a list of all the enzymes that are involved, kind of like a little summary of all the enzymes that are involved in replication. Helicase unwind, single-stranded binding proteins are going to separate. Topoisomerase, I didn't really talk about it, but topoisomerase, look at my cursor. It's going to act right here. It's going to relieve the strain upstream so DNA doesn't get like super wound up. And I have this really great demonstration I do during class where I walk up to someone who's wearing like a hoodie that they're not supposed to with little strings on it. If you wrap the strings around each other and then you pull them, it super coils. And you're probably not understanding what I'm saying right now. So all I want you to know is that topoisomerase is going to relieve supercoiling the strain up here and the risk of supercoiling. Okay. And then primase does the primer. DNA polymerase 3 is the one that comes in first and puts down new DNA. DNA polymerase 1 replaces primer. Ligase is going to seal it all together. And then there's your summary picture. Beautiful. All right, proofreading and repair. Let's talk about mistakes being made because not all enzymes are perfect, just like not all people are perfect. We make typos, and so does DNA polymerase. DNA polymerase, it's kind of like if you're typing something on your computer or laptop and you accidentally mistype and you notice and then you delete it and then you put the right letters. DNA polymerase actually proofreads itself as it goes, kind of like you typing something and correcting yourself. And so DNA polymerase is going to make, even after it corrects itself, it's going to make 1 in 100,000 mistakes. 1 in 100,000 mistakes. That's a lot. We have 3 billion base pairs of DNA. We would be totally mutated if this happened. So mismatch repair, nucleotide excision repair, other special enzymes come in and they're going to correct the mistakes that DNA polymerase missed. Okay, so I would pause right here if I were you and write this down. Special enzymes fix incorrect pairings, nucleases cut out damaged DNA, DNA polymerase and ligase come in and fill in the gaps. So it's like if you're writing a paper and you make mistakes, but you're like, oh gosh, this is so awesome. I corrected all my mistakes. I was so careful. I'm going to turn this into my English teacher. And then your English teacher gets it, and your English teacher is considered like the mismatch repair. Your English teacher is like, nope, and they put like a bunch of red marks on your paper. Then they hand it back to you and they say, all right, you need to correct this. So you would be DNA polymerase, the teacher would be the mismatch repair special enzymes, and then you would end up, being DNA polymerase again and correcting it. Hopefully that was a good analogy. I just came up with that off the top of my head. Just an FYI, you don't need to write this down, but a form of colon cancer has been found to involve genetic mutation in a nuclease. So if these nucleases don't work right, then you end up mutated, and cancer is caused by DNA mutation. So repair is an ongoing process in cells due to environmental factors causing mutation. So our nucleases are hard at work all the time. And this is what it looks like when there's cutting out of damaged or incorrect DNA and repairing of it. So remember I said that DNA polymerase makes 1 in 100,000 mistakes. After excision repair, there was only 1 in 10 billion. Whew, that's good to know. I mean, even if we didn't know it, you don't have to know it to experience the benefit, but that's a good thing. 1 in 10 billion is a good thing. You have 3 billion base pairs, so this is a good thing. All right. Last thing is you have linear chromosomes. So every time your cells divide, DNA gets chopped shorter and shorter and shorter. And this is actually something really interesting, because this is something that if anyone goes into like cancer research, they have to know about this. Because DNA polymerase can only add to the 3' end of a nucleotide, this comes up over and over again, it can only add to the hydroxyl side. It cannot replace the RNA primer that is at the very tip of the chromosome. Therefore, the chromosome gets chopped. The RNA primer just gets chopped off. All right? The very tip of a chromosome, the RNA primer gets chopped off because DNA polymerase 1 just can't do it. Just can't do it because it's like, nope, there's no 3' end for me to grab onto. I'm not doing it. So it gets chopped. That would be a problem, right? If that was coding DNA, that would be a big issue if our chromosomes are getting chopped into coding DNA. But we have this really cool thing. These things are called telomeres. So the question says, in case you're wondering, what am I supposed to write down? Why does DNA grow shorter and shorter with every replication? Because the primers at the tips of the chromosomes cannot be replaced and are therefore chopped off. We have these really cool things called telomeres. Telomeres are repeated units of nucleotide sequences at the ends of your chromosomes, repeated over and over and over and over and over. And they're non-coding. So if they get chopped off, like it's cool, whatever. The analogy that I use is these are like aglets. I don't know, I've been teaching this for like 7-8 years. And every time I say aglet, students are like, oh, Phineas and Ferb. I don't even know anymore if your age group ever watched Phineas and Ferb. I'm so sad they took it off of Netflix. My kids, we used to watch it every morning. Anyways, they're like the aglets. An aglet is in case you haven't watched Phineas and Ferb, they're like the little plastic things at the end of your shoelace that protects the shoelace from fraying. So a telomere is kind of like a little protection from coding DNA from being chopped and fraying, right? If I were to keep cutting the aglet of my shoelace, I would eventually run out of aglet and I would end up fraying my actual shoelace, which would be just catastrophic because I don't want a frayed shoelace. So what happens when our chromosomes, all the telomeres end up chopped off? What happens when the cells have divided so many times that the telomeres have been chopped off? The cell undergoes apoptosis. All right, so telomeres are like the aglets of the chromosome. They get chopped every time a cell divides. Whenever telomeres get chopped enough to where they run out, the cell undergoes apoptosis. And which organelle is involved in apoptosis? Somebody tell me. Say it out loud. Which organelle is involved in apoptosis? Which organelle releases hydrolytic enzymes? I hope you all said lysosomes. If you didn't, go back and study the cells chapter or the cells lecture. All right, and then last thing I want to leave you with is there is an enzyme called telomerase that comes in and adds telomeres back. We don't want that to happen. So telomerase would be, here's my analogy. A person, when they get old, like the older a person gets, the worse they get at driving. I'm sorry, but it's true. When they get into, oh, you should see my parents. Oh my gosh. They're in their 70s and driving is becoming an issue. So we kind of need to we're going to have to take their driver's licenses away at some point. It's going to be so crazy, but telomerase would be like if you took the driver's license away from a 75-year-old that was driving recklessly. Telomerase would be a person just giving it back. Telomerase is an enzyme that will add telomeres back. We don't want telomerase. Telomerase is an enzyme that is found in cancer cells. So if telomerase keeps adding telomeres back, that cell will never undergo apoptosis. Therefore, cancer cells are immortal, meaning cancer cells never die. If you put cancer cells in a petri dish, they'll live, like with enough nutrition, they'll live forever. And I'm going to talk about that when I talk about cancer in a couple of days. So I want you to remember that because that's something very important to me and to the topic of cancer. And with that, I think we have everything written down except here's just a picture that shows you telomerase coming back in like, oh, I'm helping adding telomeres back, but we don't want that. We need our cells to die when they get too old and when they've divided too many times. All right, I will end it there and I will talk to you guys later.